Today we'll be covering Chapter 30 that includes topics pertaining to agents of bioterrorism. This lecture was adapted from the original version created by Bhavna Desai, a part-time microbiologist at the Valley Hospital and an infection preventionist at St. Mary's General Hospital in Passaic, New Jersey. So, bioterrorism is defined as the intentional or threatened use of bacteria, viruses, fungi, or toxins from living organisms to produce death or disease in humans, animals, or plants. The purpose of bioterroristic acts are to cause civil and economic instability that leads to fear, panic, and overall chaos. Bioterrorism acts can be either overt or covert. So the overt attacks have immediate impact and also immediate emergency response, whereas the covert acts are a little more challenging as there may be no initial announcement of the attack, and it may not be recognized till a little time later. So we discussed some of these concepts in Chapter 4 regarding biosafety and the levels, but we'll revisit them since they are very applicable to this topic of bioterrorism. So the biosafety levels are categorized into 4 levels. The first one being biosafety level 1, which is suitable for working with agents that are not known to cause disease in healthy adults. Minimum safety procedures are required. So an example of an organism that would be handled in a biosafety level 1 laboratory would be Bacillus subtilis. The second level, biosafety level 2, is suitable for working with agents of moderate potential hazard. So these are ones that are known to cause human disease but are not readily transmitted. So examples of these would be the hepatitis V virus and salmonella. Also in a biosafety level 2, the biological safety cabinets or hoods are to be used. So a class 2 is recommended in this level. Biosafety level 3 is suitable for working with agents that may cause serious or potentially lethal disease due to exposure inhalation. So these are organisms that are infected via the respiratory route. So examples of these include Mycobacterium tuberculosis or Brucella. And then lastly, biosafety level 4, that is suitable for working with dangerous agents that cause life-threatening disease. So examples of these are Ebola or Marburg virus, along with the Congo Crimean hemorrhagic fever virus. In an effort to increase responsiveness and to create a nationwide ability to rapidly detect both covert and overt deployment of biologic weapons in the United States, the CDC, along with the Association of Public Health Laboratories, the FBI, and various other government agencies, established the Laboratory Response Network in 1999. The goal of the Laboratory Response Network was to decentralize testing capabilities and to link both state and local laboratories with advanced capacity clinical, military, veterinary, agricultural, water, and food testing laboratories. As defined by the Laboratory Response Network, community hospitals with microbiology capabilities are designated as Sentinel laboratories. They were formerly known as Level A laboratories. Sentinel laboratories have to have biosafety level 2 facilities and utilize laboratory protocols published by the CDC and American Society for Microbiology to rule out the presence of biologic agents. Laboratory Response Network B designated laboratories are reference laboratories, and these are capable of confirming identification of bioterrorism agents. Usually they include public health laboratories, along with the Department of Defense Medical Center laboratories. It also can include national laboratories, which are most technically capable laboratories of having the testing capable to identify these agents. So this diagram depicts the hierarchy of the laboratory response network. So at the bottom are the sentinel labs. So these are the community or hospital laboratories. And again, their main role is to rule out bioterroristic agents and then refer any suspicious isolates to the reference laboratories or national laboratories. So they should not be identifying these bioterroristic agents. They should strictly be ruling out the presence of them. In the event of a bioterror event, clinical microbiologists play a key role. Detection of possible bioterror event requires active microbial surveillance programs. Laboratory scientists need to be trained to recognize potential agents, and communication with clinicians, emergency response personnel, and infectious disease specialists are paramount. This helps communicate from the sentinel lab level to the reference labs and then at a national level. Within the laboratory response network, there needs to be a plan in place for each lab, delineating what is needed and their respective role. So there's several elements involved in the Sentinel Lab response plan that involves a relationship between the public health team, such as the government agencies, and the Sentinel Lab, the hospital or community laboratories. One important element is the involvement of management. So both the laboratory management and the hospital management need to be involved in the establishment of protocols, written policies, and training for appropriate procedures for the appropriate staff. Another element is safety. So of course, proper equipment needs to be in place for the correct biosafety level that the laboratory is, and then safety supplies such as splash guards, appropriate PPE, etc. Security is an important component because all laboratories should have limited access and only certain personnel should be allowed to be within the laboratory. Training procedures need to be in place as well. And these procedures should lay out policies of who's handling any potential bioterrorism agents, how they're being shipped out, and overall who's allowed access to them. Finally, supplies is another important element to ensure that there's adequate supplies to do routine testing and those supplies needed for rule-out testing, which may not be the supplies needed on a day-to-day basis. Also, agreements need to be made with other laboratory response network laboratories if there is a need for additional supplies to rule out potential agents. The important procedures to remember being a Sentinel lab includes that the Sentinel labs are responsible for collection of samples and preliminary testing from patients seen at the respective facility. They're then involved in detecting or isolating suspected agents according to the respective facility's protocols and ruling out the presence of such agents. If an agent of bioterrorism is suspected, they should not do full workups, as mentioned before, but work in coordination with local government and state agencies, along with public health laboratories. In doing so, they subsequently will send out or refer the samples to these reference laboratories or public health labs. In some cases, the patients may also be referred out to a designated facility. Table 30.1 in your textbook provides a more extensive list of select agents and toxins that are deemed either biologic threats or select agents by various government agencies. So we're not going to be going through every single organism because, of course, it's an extensive list, but we will revisit several of the agents of bioterrorism that we've looked at in previous chapters, including Bacillus anthracis, Brucella species, Francisella tularensis, Yersinia pestis, and Burkholderia species. So again, these organisms we'll go through are not all inclusive. but we're going to focus on these ones that we've already looked at in previous chapters. So the first potential agent of bioterrorism that we'll revisit is Bacillus anthracis. So this falls under the biological agent category because of its natural occurrence in the environment and its ability to cause disease and death. Bacillus anthracis can be easily and inexpensively cultivated and has been demonstrated to cause disease through various routes, including the respiratory tract, GI tract, and through abraded skin. Because Bacillus anthracis can also produce spores, the organism can survive very harsh conditions and remains infectious. So remember, Bacillus anthracis is a zoonotic disease that can exist in three forms: cutaneous, inhalation, and gastrointestinal. Cutaneous anthrax is the most common form of the disease, and it occurs after spores are introduced into cuts or abrasions of human skin. After 1 to 6 days, a papule will appear, and then this can progress to a 1 to 2 centimeter vesicle within the next 2 days. Once the vesicle ruptures, the necrotic lesion develops into a characteristic black eschar. The lesion usually heals without need for intervention. Oral or gastrointestinal anthrax occurs when spores are introduced into the tissue of the gastrointestinal tract, either by eating meat containing anthrax spores. There are two distinct manifestations: oropharyngeal, which is localized, or the GI presentation, which can occur anywhere within the GI tract. After incubation periods of 2 to 6 days, the patient may experience a sore throat, difficulty swallowing, and swollen lymph nodes, along with nausea and vomiting. Intestinal anthrax has a variable mortality rate because of delayed initiation of appropriate therapy. Inhalational anthrax occurs when the patient inhales Bacillus anthracis spores, and this is the form most likely to be seen in a bioterror event. The appropriate samples submitted are dependent on the form of anthrax that the patient presents with. So for cutaneous, you would be looking at vesicle fluid or swabs or biopsy of the eschar, the lesion. With inhalation, blood cultures or sputum cultures could be submitted, and with GI anthrax, blood cultures or stool as well could be submitted. So remember, going back to chapter 16, the aerobic gram-positive bacilli, Bacillus species are gram-positive rods, with Bacillus anthracis appearing as a large square-ended gram-positive rod. And it can appear singly or in chains, as in these figures here. Bacillus species form spores, as can be seen in this gram stain, in which the stain is not taken up by the spores. So you have these central to subterminal spores that are white or are not taken up by the stain. It's also important to remember, although these are gram-positive rods, as the cultures age, they may become gram-variable. So here, this figure and this figure almost appear gram-negative or gram-variable, whereas here's an example of a clearly gram-positive rod. Remember, another important description of these gram-positive rods are that they appear bamboo-like because of the way the rods chain one right after another. Important technical clues to remember when trying to rule out Bacillus anthracis include that the organism appears as large gram-positive rods, as we saw on the previous slide. On sheep blood agar, the colonies of Bacillus anthracis are non-hemolytic, and they appear large to gray and flat with an irregular margin. Sometimes the colonies are described as Medusa head in appearance. The growth of the colonies on the agar has a tenacious consistency and it holds tightly to the agar surface. And when the edges are lifted with a loop, they can stand upright without support, as seen in the figure on the right and also on the images on the previous slide. Colonies are also described as having a ground glass appearance on sheep blood agar. Bacillus anthracis isolates are non-motile and catalase positive. So organisms that fall into these categories may be a potential bioterrorism agent and should be referred out to a laboratory response network reference laboratory. The American Society for Microbiology provides detailed recommendations for ruling out Bacillus anthracis for sentinel laboratories. Important characteristics that should be noted for ruling out or referring these isolates include that the colonies are non-hemolytic, they have a ground glass appearance, and are non-motile, and of course that they're gram-positive rods with spores. The guidelines will be posted in Moodle for you to reference and see an example of the detailed protocol and policies for ruling out these suspected bioterrorism agents.
The next genus we'll discuss that is considered a potential agent of bioterrorism is Burkholderia. It contains two species that are considered these agents, Burkholderia malay and Burkholderia pseudomalay. Burkholderia pseudomalay is the causative agent of melioidosis. This is an aggressive granulomatous pulmonary disease transmitted by ingestion, inhalation, or inoculation of the organism. Burkholderia malay causes the disease known as glanders, and this is a disease of animals that's transmitted to humans. Usually persons who have occupations that involve contact with infected horses, mules, and donkeys, and laboratory scientists have been reported with infection. The disease manifests in three forms: a chronic pulmonary form with cough, producing a mucopurulent sputum, a form characterized by abscesses of the skin, subcutaneous tissues, and lymphatics, and an acute septicemic form that rapidly progresses from fever, chills, and prostration to death within 7 to 10 days. Glanders is not transmitted from person to person. Both Burkholderia species are transmitted by animal contact, but they're endemic in different parts of the world. So Burkholderia malay is usually found in Asia and several Eastern Mediterranean countries, whereas Burkholderia pseudomalay has a similar disease presentation, but it's found in different parts of the world. It's endemic in Southeast Asia, particularly Thailand, the South Pacific, Africa, India, and the Middle East. Similar to other potential bioterrorism agents, laboratory transmission is a high risk with culture and manipulation of Burkholderia. Physicians suspecting infections by Burkholderia malay or pseudomalay should notify the clinical laboratory when submitting specimens for culture. Specimens appropriate for submission to detect either of these isolates are blood, bone marrow, sputum, bronchoalveolar lavage, abscess material, urine, and serum. Throat or nasal swabs can also be submitted. Direct gram staining of aspirates and respiratory secretions for suspected Burkholderia infection reveal gram-negative cocobacilli for Burkholderia malay and small gram-negative bacilli for Burkholderia pseudomalay. These organisms can also be noted on direct exam of blood cultures from suspected patients. Cultures should be incubated at 35 degrees Celsius and 5% CO2 for up to 5 days. Burkholderia malay grows as small, circular, butter-like colonies after 24 hours to 48 hours of incubation on sheep blood agar. but if held longer, the colonies can convert to dry, wrinkly appearance, and these will actually resemble Pseudomonas stutzeri. Burkholderia malay and Pseudomallay typically will grow on McConkey agar. And one differentiating characteristic between the two is that Burkholderia pseudomallay is described as having a soil-like smell when the bacteria grows in culture. But of course, as we've discussed previously, it's hazardous and it should never be attempted to smell an open plate directly. Clues for ruling out Burkholderia species include observing gram-negative cocobacilli or small gram-negative bacilli with pore growth at 24 hours, forming larger non-pigmented gray colonies at 48 hours that are indole-negative, non-motile, catalase-positive, and resistant to polymyxin B or colistin. These can be presumptively identified as Burkholderia malay and must be submitted immediately to the nearest laboratory response network reference laboratory. Gram-negative cocobacilli or small gram-negative bacilli that exhibit pore growth at 24 hours but form larger non-pigmented gray colonies at 48 hours and that begin to wrinkle after 48 hours will grow in McConkey, are oxidase positive and indole negative, and are resistant to polymyxin B or colistin, may be potentially a Burkholderia pseudomallay isolate. An isolate with these characteristics should also be referred to the Laboratory Response Network reference laboratory for confirmation. Tularemia is a plague-like zoonotic disease caused by Francisella tularensis. And this is the next agent of bioterrorism that we'll discuss, and another organism that we've already looked at in previous chapters. Human infections usually occur by accidental contact with the organism, by an arthropod bite, or by direct contact with infectious material, ingestion of contaminated food or water, or by inhalation of infective aerosols. As with the other potential agents of bioterrorism, this organism is dangerous and highly virulent, and it should not be manipulated on an open bench. And again, as with the other organisms in the Sentinel laboratory, we're trying to rule these organisms out and not fully identify them. Since the infectious dose for this organism causing cutaneous or inhalational disease is very low, it's really important to be familiar with the test to rule it out. And there have been documented cases of laboratory-acquired infections. Francisella tularensis can be observed in direct specimen smears, and appropriate specimens include blood culture, wound biopsies or swabs, lymph node aspirates, and sputum or bronchial washings. On gram stain, Francisella appears as tiny pleomorphic gram-negative cocobacillus. It's a fastidious organism requiring the amino acid cysteine for optimal growth, so it will grow well on media supplemented with cysteine such as chocolate agar, modified Thayer Martin, cysteine hard agar, and buffered charcoal yeast extract agar. Francisella grows very slowly, so colonies may not be visible initially on solid media until after 36 to 48 hours of incubation. It will not grow on McConkey agar or eosin methylene blue agar. Pleomorphic, poorly counterstaining gram-negative cocobacilli from blood cultures that do not grow well, if at all, on sheep blood agar should be a clue or an indication to the laboratory scientist to consider Francisella tularensis. Laboratory procedures that should be done must be carried out in a biological safety hood until it's ruled out. Important tests to include are catalase and beta-lactamase, which are positive for Francisella tularensis, whereas X and V requirements, urease and oxidase, are negative. It's important to note that the catalase may be a weak positive reaction, and then the reason for observation of X and V or satellite testing is to rule out Francisella versus Haemophilus species. So when these tests are completed and isolates may be suspicious, again, they should be referred to the Laboratory Response Network reference laboratory for confirmatory identification, and usually this is PCR.
The next significant organism is Yersinia pestis. So this is the causative agent of the plague. It's extremely pathogenic and exists in three forms. There's the pneumonic, the bubonic, and then the septicemic. So the pneumonic form infects the lungs. The bubonic infects the lymph node glands, and they appear swollen and tender. This form does not spread from person to person. And then there's septicemic, in which bacteria multiplies in the blood. Yersinia can be isolated from numerous samples and observed on direct gram smear. The sample sources include blood culture, lymph nodes, aspirates, respiratory cultures, cerebral spinal fluid, and also scrapings from the lesions. Yersinia pestis has a distinct gram stain appearance. It resembles bipolar safety pins and stains as a gram-negative bacilli. The colonies resemble Anthrax when they grow, but they grow better at 28 degrees rather than the normal 35 degrees. Yersinia pestis is a non-lactose fermenter on McConkey plates. It's catalase positive, oxidase negative, indole negative, and urease negative. Isolates of this organism, similar to the others, would be referred to the laboratory response network. The image at the top is showing a right stain of Yersinia pestis from a blood sample of a plague victim. Note the darker stained bipolar ends of the organism that result in the classic safety pin appearance. On the bottom, the figure of the blood agar plate shows Yersinia pestis in culture after 72 hours incubation. The colonies appear gray-white to slightly yellow opaque, and they're raised with an irregular fried egg appearance. Alternatively, there's colonies that may have a hammered copper, shiny surface as well. Brucella species is the last organism that we'll discuss that's an agent of bioterrorism. So this is the most commonly reported laboratory-associated bacterial infection. Infections often occur by sniffing cultures, direct skin contact, and then aerosol-generating procedures. So Brucella can be observed in direct smear, and the specimen sources include blood or bone marrow, and then tissue samples such as spleen or liver. Upon gram stain, Brucella appears very tinily and stains faintly, but it's a gram-negative cocobacilli. It's a slow grower on sheep blood agar and chocolate, and colonies appear 2 to 3 days after incubation. It's a catalase positive organism, oxidase positive, urease positive, and nitrate positive. And again, this organism would be referred to the laboratory response network if a suspected isolate was found. There's a couple helpful hints for detecting bioterrorism agents. So number one, if an agent is suspected, inoculate and incubate extra sets of blood agar, McConkey, chocolate, and thioglycolate broth at 28 degrees, 35 degrees, and 42 degrees Celsius. Only Yersinia grows at 28 degrees, but it resembles some Anthrax, so that's a way to rule them out. And then only Burkholderia studimale grows at 42 degrees. Another helpful hint is growth on McConkey. So Yersinia is a non-lactose fermenter. Burkholderia malae is a lactose fermenter and appears light pink after 72 hours. And Burkholderia studimale is a lactose fermenter after 24 to 48 hours, and the colonies appear dry and wrinkled after 48 to 72 hours. Urea is helpful in identifying Brucella, which would be urea positive. And then beta-lactamase is helpful with Francisella, with that being a beta-lactamase positive organism. Aside from the organisms mentioned on previous slides, there's other possible agents of bioterrorism, and they include toxins, viruses, and then of course other bacteria that were not mentioned. So toxins can include botulinum toxin, Staphylococcus aureus, enterotoxin B, and ricin. Viruses include variola major, the causative agent of smallpox, the Marburg virus, which causes hemorrhagic fever, Ebola virus, and Venezuelan equine encephalitis. Additional bacteria include Coxiella burnettii, but then remember the organisms we discussed are not all inclusive of all the possible agents of bioterrorism. But with that, any of these organisms would not be processed if specimens were suspected of infection or contamination with these agents. Once a bioterrorism agent is suspected or isolated, the next very important step is reporting. So in the Sentinel labs, the first thing would be to notify a supervisor, a manager in the microbiology lab, and then escalate it to a laboratory administrative director. Along with the laboratory administration, hospital administration also needs to be notified. So that would include infection control or the infection preventionists, and also infectious disease doctors. After reports are given to internal contacts, then the agents of bioterrorism need to be reported to outside agencies as well. So agencies such as the Communicable Disease Service, CDRSS, that's a Communicable Disease Reporting and Surveillance System, and the local Board of Health and possibly the FBI all need to be notified if there's agents of bioterrorism suspected. So the next important step in the laboratory response plan is referring isolates after suspected cases have been reported. So when a select agent cannot be ruled out within the Sentinel laboratory protocol, then the laboratory is to cease identification procedures and refer the confirmatory identification of the agent to the next laboratory response network tier, which would be the reference laboratories. In New Jersey, that's the Public Health, Environmental, and Agriculture laboratories that are part of the New Jersey Department of Health. Once a suspected isolate is reported and referred, it will need to be shipped to the Public Health Laboratory. So there's specific guidelines for shipping and packing. Also a chain of custody form. It's the Lab 5 form. The steps include calling the Communicable Disease Service and obtaining a case number before filling out the form. The isolate should be packaged as infectious according to the Department of Transportation, and also a shipper's declaration needs to be filled out. The Lab 5 form is filled out as per the State Public Health Laboratory, and that is attached to the sample to be shipped. The chain of custody form includes the name, address, phone number of the originating laboratory, the reason for the transfer of the isolate, who released the isolate, the name and the signature, the date and time of transfer, and then who received the isolate, the name, signature, agency, and phone number. Transportation to the Public Health, Environmental, and Agricultural laboratories is usually conducted by the state laboratory. So typically a courier or a state employee will come to the laboratory to pick up the isolate to transport it appropriately. So now that we've gone through the laboratory response network, the possible plans, some of the possible agents of bioterrorism, there's also a lot of informational materials and resources available online. The Federal Emergency Management Agency, also known as FEMA, that has an independent study program available online. It's for U.S. citizens only, but their Emergency Management Institute offers self-paced courses that are designed for people who either have emergency management responsibilities that could include laboratory personnel, and then also courses for the general public. So all these are offered free of charge to those who qualify for enrollment. So down below you'll see there's just directions on how to access the courses. So you click on the ISP courses from the menu and then select college credit from the box on the right if you choose to complete any of these courses. This slide provides resources for regulations regarding the possession, use, and transfer of select agents and toxins. The CDC had reviewed the list of biological agents and toxins that had the potential to pose a severe threat to public health and safety and had proposed to amend and republish the list in 2010 to 2011. An executive order had also been signed to designate a subset of the select agents and toxins list that presents the greatest risk of deliberate misuse, with the most significant potential for mass casualties or devastating effects. And finally, this last slide provides some additional resources for information on agents of bioterrorism and training resources. So the CDC has an emergency cdc.gov website that has specific information on bioterrorism. The Association of Public Health Laboratories, also known as APHL, has resources on their website, along with the American Society for Microbiology that has specific guidelines for sentinel laboratories.